Parkinson's disease is the second most common neurodegenerative disease, affecting approximately 10 million people worldwide. The greatest risk factor for developing Parkinson's disease is age, with prevalence rates roughly doubling every 5 years for both men and women between the ages of 50 and 69. In the UK, it is estimated that 1 in 37 people will receive a diagnosis of Parkinson's disease during their lifetime. However, by 2025, the frequency of this condition is expected to rise by 20% due to population growth and the increasingly aging population. Parkinson's disease is classically defined as a motor disorder. Often, it is easy to spot individuals with the disease, as they will present with unique motor symptoms demonstrated by this animation. These symptoms include tremor or shaking, rigidity, a slow shuffling movement, and balance problems. But what is it that causes these problems? These motor symptoms are a product of cell death in regions of the brain that control movement, most importantly, dopamine-containing neurons in the nigrostriatal tract. Sections of the brain that are home to these dopamine-containing neurons are very easily identified, as these neurons have a distinctive black colour. In the brain of people with Parkinson's, however, these dopamine-containing neurons are lost, and the areas of the brain that mediate movement lose their characteristic black pigment. Death in these neurons is primarily a result of accumulation of a misfolded protein called alpha-synuclein. Alpha-synuclein is a protein found in neurons and is believed to play a role in neurotransmitter release. It is not quite clear what triggers misfolding of alpha-synuclein, but it is likely that either genetic mutations or exposure to harmful chemicals in the environment may cause the initial damage to the protein. In healthy cells, there are systems that exist to remove misfolded and potentially harmful proteins from cells, but in Parkinson's, these systems fail, which allows misfolded alpha-synuclein to accumulate within the cell. As more and more alpha-synuclein accumulates in neurons, it starts to form a pathological structure known as a Lewy body. Lewy bodies are crucial in the diagnosis of Parkinson's disease, as their location across the brain is closely related to the clinical presentation of the disease. Lewy bodies are capable of interfering with the neuron's normal function and can even promote cell death. These proteins are believed to be transferred between neighbouring neurons, which explains the gradual spread of neuronal damage across the brain, and the diverse symptoms of Parkinson's disease. Lewy bodies originate in the brainstem and the olfactory bulb, which leads to degeneration of serotonin-containing neurons that mediate mood. This results in symptoms of anxiety, depression, and even loss of taste or sense of smell. Interestingly, these symptoms can occur up to 10 years before motor symptoms even start to develop. From here, the Lewy bodies eventually spread to the areas of the brain that mediate motor control, which leads to the development of the widely recognized motor symptoms of Parkinson's disease. In the late stages of the condition, Lewy bodies continue to affect the areas of the brain that mediate movement, but also progress into the cortex, which primarily affects cognition and leads to the development of dementia, depression, and psychosis in individuals with Parkinson's disease. So how do we treat Parkinson's disease? Most of the available treatments actually work to eliminate the motor symptoms and don't really tackle any of the non-motor symptoms, such as anxiety and depression. An important category of drugs used to combat these motor symptoms are the drugs that replace dopamine lost in the brain. If you can recall earlier, we described how these motor symptoms arise in patients with Parkinson's, and this was due to the loss of dopamine-containing neurons in the nigrostriatal tract, a region of the brain that controls movement. We are now going to zoom in on these dopamine-containing nerves that are crucial for motor control so that we can understand how drugs used to manage the motor symptoms of Parkinson's disease actually work. Here we have a gap between two dopamine-containing neurons. Neurotransmitters such as dopamine act as signals and are transferred from one neuron to another to allow for communication between the neurons. In this case, the signals transferred in the nigrostriatal tract are important for motor control. In the neuron, dopamine is synthesized from a precursor chemical known as L-dopa. Enzymes in the neuron create dopamine from L-dopa. And here, dopamine is packaged into these small circular structures that act as small stores of dopamine. These dopamine-filled stores then migrate to the end of the neuron and release dopamine into the gap between the two neurons. Dopamine can then bind to proteins on the surface of the neighbouring neuron and trigger the same sequence of events. Once dopamine has activated the neighbouring neuron, it is removed and taken up into nearby cells or degraded by enzymes outside of the neuron. Naturally, as dopamine-containing neurons die in the nigrostriatal tract, there are fewer signals between neurons, which impairs motor control in people with Parkinson's disease. So, in an effort to manage the motor symptoms, it would make sense to increase dopamine signaling between neurons, and this is what most of the approved drugs for Parkinson's disease do. The gold standard drug for Parkinson's is simply the precursor chemical of dopamine, otherwise known as L-dopa. When administered to patients, L-dopa is converted to dopamine in neurons, which increases dopamine signaling. Another class of drugs used to treat Parkinson's disease limit degradation of dopamine, again increasing dopamine signaling across the brain. Or other drugs simply mimic dopamine in the brain by activating the same proteins on the surface of neurons. While these drugs work to reduce the motor symptoms of Parkinson's disease, they do very little to combat the non-motor symptoms, which are in some cases equally disabling. An important limitation of current treatments for Parkinson's disease is the undesirable effects these drugs often have on patients. Dyskinesias are an unfortunate, often painful side effect of these drugs, characterized by uncontrolled, involuntary movements that are usually localized to the face. Unfortunately, dyskinesias occur in 80% of people with Parkinson's disease 5 to 10 years following treatment with L-dopa, the gold standard drug for Parkinson's. Therefore, there is a clear need for the development of new drugs to treat Parkinson's disease. There are no currently available disease-modifying treatments for Parkinson's disease, which is a concern given the increase in the prevalence of the disease and the aging population. Research in this field points towards treatments that target misfolded alpha-synuclein, the believed perpetrator of neuronal damage in Parkinson's. At the moment, there is a drug that has been proven to help clear damaged alpha-synuclein in stage 3 clinical trials, which sounds promising. So hopefully in the next few years, we will see a considerable rise in the number of treatments available to people with Parkinson's disease that not only target the motor symptoms, but limit disease progression and alleviate the disabling non-motor symptoms of the disease.